Hello friends, this is the second video of microscopy and morphology of bacteria. So let's begin from bacterial anatomy. The outer layer of cell envelope of a bacterial cell consists of two components. First is a rigid cell wall, second is the underlying cytoplasmic or plasma membrane. The cell envelope encloses the protoplasm, which comprises cytoplasmic inclusions, inclusions including mesosomes, ribosomes, inclusion granules, vacuoles, and a single circular chromosome of deoxyribonucleic acid, that is, in short, DNA. Besides these essential components, some bacteria may possess additional structures such as capsule, flagella, and fibriae. Now let's know about cell wall. The cell wall is a tough and rigid structure surrounding the bacterium like a cell. It is 10 to 25 nanometer in thickness and weight about 20 to 25% of the dry weight of the cell. The cell wall has some functions. First is accounts for the shape of the cell. Second is provides protection to the cell against osmotic damage. Third is confers rigidity upon bacteria. Fourth is it takes part in cell division. It possesses target site for antibiotics, lysozymes and bacteriophages. It carries bacterial antigens that are important in virulence and immunity. The rigid part of the cell wall is a peptidoglycan, which is a mucopeptide, that is, in short, murin, composed of N-acetyl muramic acid and N-acetyl glucosamine molecules alternating in the chains cross-linked by peptide subunits. The cell walls of gram-positive bacteria have simpler chemical nature than those of gram-negative bacteria. Now let's know about the gram-negative cell wall. The gram-negative cell wall is a complex structure with some components. Let's know about these components. First is lipoprotein layer. It connects the peptidoglycan to outer membrane. Second is outer membrane, that is, this contains certain proteins named as outer membrane proteins. These are target sites for phages, antibiotics, and bacteriocins. Lipopolysaccharides. Let's know about the third component, that is lipopolysaccharide. This layer consists of lipid A, to which is attached a polysaccharide. LPS is lipopolysaccharide, constitutes the endotoxin of gram-negative bacteria. The polysaccharide determines a major surface antigen, the O antigen. The toxicity, that means pyrogenicity, lethal effect, tissue necrosis of bacteria is associated with lipid A. The periplasmic space. Fourth one is the periplasmic space. It is the space in between the inner and outer membrane. It contains various binding proteins for specific substrates. Fifth is peptidoglycan. Now let's know about gram-positive cell wall. Gram-positive cell wall. Under this, first we will learn about peptidoglycan. In peptidoglycan, this layer is gram-positive bacteria is thicker, that means 16 to 80 nanometer, than that in gram-negative bacteria. In gram-negative bacteria, it is 2 nanometer. In gram-positive bacteria, it is 16 to 80 nanometer. Second is teichoic acid, that is in gram-positive cell wall. In gram-positive cell wall contains significant amount of teichoic acid, which is absent in gram-negative bacteria. The teichoic acids constitute major surface antigens of gram-positive bacteria. They are water-soluble polymers containing ribitol or glycerol polymers. Other components. Let's know about the other components. Certain gram-positive cells Also contains antigens such as protein and polysaccharides. Now let's know about the demonstration of cell wall. The cell wall contains the cell wall cannot be seen by light microscope and does not stain with simple dye. Demonstration of cell wall can be done by methods such as plasmolysis. When bacteria is placed in a hypertonic saline, shrinkage of cytoplasm occurs while cell wall retains original shape and size. Now let's know about dissection. Microdissection. Second and third is differential staining. The fourth is reaction with specific antibody. Fifth is electron microscopy. Now let's know about bacteria with defective cell wall. The synthesis of cell wall may be inhibited or interfered by many factors such as antibiotics, bacteriophages, lysozymes. Lysozyme and Lysozyme is an enzyme normally present in many tissue fluids. Lyses susceptible bacteria by splitting the linkage of peptidoglycan in the cell wall. When lysozyme acts on a gram-positive bacterium in a hypertonic solution, a protoplast is formed. In case of gram-negative bacteria, the result is a spheroplast. Bacteria with defective cell wall may probably have a role in the persistence of certain chronic infections such as pyelonephritis, and bacteria without cell wall or with deficient cell walls are of four types. First is mycoplasma, L forms, protoplast and spheroplast. Now first we will study about mycoplasma. This is a naturally occurring bacteria without cell walls. Mycoplasma is classified as an independent bacterial genus. They do not require hypertonic environment for maintenance and are stable in culture medium. Now let's know about L form. Klinberg, Klinberger Nobel, while studying Streptobacillus moniliformis in the Lister Institute, London, observed abnormal forms of bacteria and named them L forms after the Lister Institute. L forms develop either spontaneously or in the presence of penicillin or other agents that interfere with synthesis of cell wall. These are difficult to cultivate and require agar containing solid medium having right osmotic strength. L forms are sometimes spontaneously formed in patients treated with penicillin. L forms are more stable than protoplast and spheroplast. Now let's know about protoplast. These are derived from gram-positive bacteria. They contain cytoplasmic membrane and cell wall is totally lacking. These are produced artificially by lysozyme in a hypertonic medium. These are unstable. Hypertonic condition is necessary for their maintenance. Now, let's know about spheroplast. These are derived from gram-negative bacteria. They are produced in presence of penicillin. They are osmotically fragile and must be maintained in hypertonic culture medium. They differ from the protoplast in that some cell wall material is retained. Now let's know about pleomorphism and involution forms. Certain species of bacteria exhibit great variation in shape and size of individual cells called pleomorphism. Some bacteria show swollen and aberrant forms in aging laboratory cultures and are known as involution forms. Defective cell wall synthesis is often responsible for development of these two abnormal forms. Now let's know about cytoplasmic membrane. It is 5 to 10 nanometer thick, elastic, semi-permeable layer which lies beneath the cell wall, separating it from the cell cytoplasm. Electron microscopy shows presence of three layers constituting a unit membrane. Chemically, it consists of lipids and protein molecules. Sterols are absent except in mycoplasma. Cytoplasmic membrane acts as an osmotic barrier. It is the site of numerous enzymes, that means formages, oxidases, and polymerases involved in the active transport of selective nutrients. It acts as a semi-permeable membrane controlling the inflow and the outflow of metabolites to form the protoplasm. It also contains cytochrome and enzymes necessary for the cell wall synthesis. Now let's know about cytoplasm. The bacterial cytoplasm is a colloidal system containing a variety of organic and inorganic solutes in a viscous watery solution. It lacks mitochondria and endoplasmic reticulum of eukaryotic cell. It contains ribosomes, mesosomes, vacuoles, and inclusions. The cytoplasmic stains uniformly with basic dyes in young cultures. Now let's know about ribosomes. These are the centers of protein synthesis. These are composed of ribosomal RNA, that means in short rRNA, and ribosomal proteins. Ribosomes are integrated in linear strand of mRNA to form polysomes, and it is at this site that code of mRNA is translated into peptide sequence. They are 10 to 20 nanometer size with sedimentation constant of 70 S. That means S stands for suburban Borg units. Now let's know about intracytoplasmic inclusions. These are sources of stored energy and present in some species of bacteria. Their function and significance are uncertain. They may be present as polymetaphosphate, that means volutin, lipid polysaccharide, lipid and polysaccharide, and granules of sulfur. They are most frequent in bacteria grown under conditions of nutritional deficiency and disappear when the deficient nutrients are supplied. Now let's know about mesosomes. Chondroids. Mesosomes are vesicular, multilaminated or convoluted structures formed as invaginations of the plasma membrane into the cytoplasm. They are the principal centers of respiratory enzymes and are analogous to mitochondria of eukaryotes. These are more prominent in gram-positive bacteria. There are two types of mesosomes, septal and lateral. The septal mesosomes is attached to bacterial chromosome and is involved in DNA segregation and in the formation of cross walls during binary fission. Now let's know about nucleus. Bacterial nucleus has no nuclear membrane or nucleolus. It cannot be demonstrated by ordinary microscope but needs an electron microscope. The nuclear deoxyribonucleic acid does not appear to contain any basic protein. The genomic DNA is double-stranded in the form of a circle. It measures about 1 mm, means 1000 micrometer when straightened. The bacterial DNA is haploid, replicated by simple fission, and maintains bacterial genetic characteristics. Some bacteria may possess extranuclear genetic material in the cytoplasm consisting of DNA named as plasmids or episomes. The plasmid replicates autonomously. They are not essential for the life of the cell but may confer on the bacteria certain properties such as drug resistance and toxigenicity, which constitute a survival advantage to the bacteria. These plasmids can be These plasmids can be transmitted from one bacterium to another either by conjugation or by the agency of bacteriophage. Besides these methods, plasmids may be transferred to daughter cells during cell division by binary fission. Now let's know about bacterial capsule and slime layer. It is the amorphous viscid bacterial secretion which surrounds some bacteria as their outermost layer. When it diffuses into surrounding medium and remains as a loose on undemarcated secretion as in leuconostoc, it is known as slime layer. When this secretion is organized into a sharply defined structure as in Streptococcus pneumonia, it is known as the capsule. Capsules which are very thin and cannot be demonstrated under the light microscope are called macrocapsules. Example, Neisseria meningitis. The slime layer or capsule is generally polysaccharide in nature, but it is polypeptide in anthrax bacillus. Some bacteria may have both a capsule and a slime layer. Bacteria of slime produces mucoid growth on agar with a stringy consistency when touched with the loop. Now let's know about functions. Capsule enhances bacterial virulence by inhibiting phagocytosis. Loss of capsule may render the bacterium avirulent. The bacteria tend to lose capsules on repeated subcultures. It acts as protective covering against antibacterial substances such as bacteriophages, phagocytes, enzymes. Capsular antigen is specific for bacteria and can be used for identification and typing of bacteria. Now let's know about capsulated organisms. Streptococcus pneumonia, Klebsiella species, Bacillus anthracis, Haemophilus influenza, Cryptococcus neoformaminis, a fungus. Now let's know about demonstration. Now let's know about demonstration of capsule. Capsule has light affinity for basic dye, therefore it cannot be stained by gram staining. There are some methods that have been demonstrated for the capsule. First is India ink staining, that is negative staining. Capsule appears as a clear hollow round bacterium as the ink cannot penetrate the capsule. Next, second point is serological method. Capsular material is antigenic and can be demonstrated by mixing it with a specific anticapsular serum. When a suspension of capsulated bacterium is mixed with its specific anticapsular serum and examined under the microscope, the capsule appears swollen due to increase in its refractivity. This phenomenon is called as capsule swelling reaction or Quellen phenomenon. This is also named as Neufeld reaction after the person who described it. This was widely employed for the typing of pneumococci. Third is special capsule staining. This technique employs copper salts as mordants for staining of capsule. Now let's know about flagella. Flagella are cytoplasmic appendages protruding through cell wall. These are thread-like structure composed of a protein, that is flagellin, 5 to 20 micrometer in length and 0 to 0.1 to 0.02 micrometer in diameter. They are organ of locomotion. All motile bacteria except spirochetes possess one or more flagella. Now let's know about the parts and composition. There are three parts. is in flagellum. First is filament, second is hook, third is basal body. The filament lies external to the cell and is connected to the hook at the cell surface. The hook basal body portion is embedded in the cell envelope. The basal body contains outer and inner rings by which the basal body is attached to the cytoplasm membrane. Outer ring are absent in gram-positive bacteria. The flagella is made up of protein, that is flagellin, which is similar to myosin, although chemical composition of different genera of bacteria is similar. They are antigenically different. Specific flagellar antibody are produced in high titers in response to antigenic stimulation of flagella. These antibodies are useful in serodiagnosis but are not protective.